So anyway, you heard my disclaimer, and I'm totally honored to be part of Scorch and to be presenting today. This is completely different than the talk you just heard. I am taking the perspective that I'm a neurologist by training, and I think it's interesting and important to think about what we know about how HIV affects the brain and how that relates to patients. So in fact, I'm going to start out with a clinical case because I'm actually a clinician as well as an investigator, and tell you about a patient that I saw in my clinic who is a middle-aged man, I'll say at age 62, who came in with some problems with his walking and also problems with cognition. And this had happened gradually over a couple of years. And he came into the clinic with two different pictures of his brain, shown here from two different dates. And what you'll see are there's some hazy abnormalities in the subcortical white matter in his brain that are progressive over about a year and a half. So the history of this progressive change over time, and then the imaging evidence of changes in his white matter are really something that sound like the kind of time course in the clinical presentation of what we call HIV-associated dementia. However, this is a patient who had actually been on treatment for over 20 years. And this is the kind of treatment that suppresses the virus replication in the blood, that allows people to live for a very, very long time, almost a normal lifespan, living with HIV these days. But it seemed to me that there was still something happening in his nervous system. And so I think that this is really the starting off point for this talk, which is one of the really key questions in HIV at this stage, in this era of treatment, is why there are still perturbations in the nervous system and in the brain, despite the fact that we have life-saving antiretroviral therapy, which controls replication of the virus. And so the rest of the talk is basically thinking about how do we understand this. And again, for those of you who do not have a clinical background, this is really the history of HIV in the brain in 3 simple slides. So first of all, it was recognized very, very early in the pandemic, essentially as early as HIV was first described clinically, that there was a specific neurologic presentation that occurred in people with untreated HIV. And the beginning of this presentation was related to sort of a slowness and a change in behavior, difficulty with concentration. And then as this progressed and people went along, people became globally demented. And this was a terrible, terrible thing. And again, training in the early 1990s as a clinician, as a medical student, we saw lots of people in their 30s and 40s who were coming in with dementia. And some of the neuropathologic changes are shown here on the slide, showing white matter pallor, as well as microglial nodules and microglia proliferation. So lots of inflammation and local infection of brain cells by HIV. And the bottom shows a representative MRI from a patient with these kinds of changes, showing progressive atrophy of the brain, so loss of neurons, as well as changes in the white matter. So the next step of the story is the great news that one combination antiretroviral therapy, which is the current mode of therapy that uses multiple different drugs with different modes of action to successfully control the virus without developing resistance to the medications, was introduced in 1996. There was a dramatic decline in the serious non-CNS diseases associated with HIV and AIDS, as well as the brain diseases. And the brain diseases here are mostly represented by a decline in HIV dementia. The problem is, and this is data from the Charter study, which is now very ably led by Scott Latender, who I think is attending the call, and others, actually has described the fact that despite treatment, there are still persistent abnormalities of the nervous system. And this is one type of assessment which looks at cognitive testing, where standardized examinations of things like processing speed and motor function, memory, are given to people living with HIV and given to controls who are matched by age and other parameters. And what they found is only about 50% of the people had what they termed as normal cognitive function. And a swath of the rest of the people had impairment, either asymptomatic, but abnormality on testing, or actually symptomatic impairment. There's only a very small percentage of people who still have overt dementia, but there's cognitive and neurologic impairment that's ongoing in people living with HIV, even in the setting of treatment. So what do we know about CNS-related injury? How does the virus affect the nervous system? And again, this is a total oversimplification that summarizes the work of many people on this call and others. But this is just a way that I like to think about this, that basically we know that the virus circulates in the blood and mostly circulates in the blood within cells, within CD4 T cells and within potentially also monocyte cells. And that the virus is carried into the nervous system probably, and I think most evidence suggests it's carried into the nervous system within these immune cells. So these immune cells that traffic into the nervous system carry the virus in. Once they enter, they release pro-inflammatory signals that, of course, induce a cascade of more immune cell entry. So many of these are chemokines and cytokines that invite in more T cells and more monocytes. The local cells of the nervous system, the resident microglial cells, which are represented here in this yellowish cell, as well as tissue macrophages, become activated by these pro-inflammatory signals. And the damage of it, and also there can be blood-brain barrier breakdown. The neurons themselves are not infected by the virus. The virus seems to mostly infect microglial cells, macrophages, and I'll argue now it looks like T cells, which are not represented here as local CNS cells necessarily, but probably are hanging out in very small numbers. The virus doesn't infect the neurons, but the neurons are affected by the process of this microglial and this activation and inflammation, and also probably by the production of HIV proteins such as GP120 and TAT. Typically, the neurologic dysfunction associated with HIV can be reversed by the initiation of treatment. When viral replication is controlled, much of this inflammatory cascade is also dampened, and neurons can kind of rebound in their function. However, if this kind of damage goes on long enough, you actually have neuronal death, and so you have some permanent irreversible neuronal injury. So this is really a process that's kind of a bystander effect of local inflammation and local CNS infection. One of the other key questions in HIV is how reservoirs for HIV are established throughout the body. So this is a virus that integrates its genome into the host genome and stays in the body forever. And we've known for a long time that that's true in CD4 T cells that inhabit, for example, the lymph nodes of the gut-associated lymphoid tissue. But a really key question is whether or not there's local infection of brain cells and perhaps different types of brain cells that can harbor long-term infection. And one of the ways that the field has looked at this is by sequencing the viral genome of the virus that's detected in the blood, and then the virus that I'm showing here in blue that has mutated over time that's detected in the nervous system. And this is called viral compartmentalization, when unique viruses are found in the CNS compartment that are different from the blood. And this is a kind of complicated looking phylogenetic tree that basically associates the sequences of the virus found in the plasma or the blood that are pink and orange and red, and compares the sequences to those found in the cerebrospinal fluid, the fluid that surrounds the brain and the spinal cord. And has an exchange with the brain. And what you can see in this example is that there's a large cluster of viral sequences that are only found in the CNS compartment in this individual who went on for a number of times, had a number of paired lumbar punctures and blood draws that demonstrated the local sequestration of unique viral variants in the CNS and actually evolution over time in that compartment. So what this suggests is that this set of viruses is being produced from brain resident cells or CNS resident cells. And in this case, further type of virologic assessment suggested these were coming from macrophage type cells or myeloid cells in the CNS. So what I'm going to talk about for the rest of the talk are sort of two major points. One is that when in the course of infection with HIV are these processes initiated? This local inflammation, this local compartmentalization where CNS cells are actually infected by HIV. And the next question is, how do we assess these issues during treatment, focusing mostly on live humans? And thinking about this question of CNS invasion and studying live humans, I just wanted to give you a snapshot of the kinds of studies that we and many others on this call, the types of tools that we use to study live patients and study the nervous system. And this includes neuroimaging methods such as MRI imaging, lots of different modalities one can use to understand the biology of what's happening in the brain. You can use PET imaging, which tells you a variety of other kinds of sort of more functional and cellular processes. And then you can actually do a lot of studies of cerebrospinal fluid, which is much of what I'll be talking to you about today, that is not the same as looking at the brain, but it does give you a window into what's happening in the nervous system in a live patient, because this can be sampled over a variety of different time points and compared to other tissues such as blood. And the first section of this talk is talking sort of about the establishment of these pathogenic processes in the nervous system. And much of what I have learned and what we've learned in the field has come from studies of early HIV infection. I'm showing you here the photos of 3 different Scorch investigators that are also involved, highly involved in these collaborative studies that I'm going to describe. And this is a study, for example, that's focused on acute HIV infection, when people have very, very early detection of the virus in the blood without even having mounted an antibody response. So these are people who are being identified within the first few weeks of getting HIV. And this is a unique cohort that has actually been established in Bangkok, Thailand. It's a really special group of individuals. This cohort happens to be mostly male, and that's a young cohort of people who are getting HIV infection. And the studies that we have initiated there and been involved with are studying people as early as 19 days after they've acquired HIV. And what we've been lucky to do since the beginning of the cohort has been to include neurologic assessments in the study of these people. This includes lumbar punctures, collection of spinal fluid over time, and MRI imaging. A sort of parallel study that's a little different is a study that I and other colleagues ran over a period of time when people were identified with having HIV but didn't start treatment right away, which is not the current practice and not what's true in that Thai cohort. But this is a study that actually allowed us to look at the natural history over the early years of HIV and what's happening in the nervous system. So the data that I'm going to show you is actually combining both of these studies together. And the first question that really interested us was, when does HIV invade the cerebrospinal fluid, thus gaining access to the central nervous system? And this figure shows the blood viral loads in each individual person enrolled in either the Thailand study or the natural history study over time. So this is a snapshot of their initial visit and their blood viral load in red. And superimposed on that, I'm showing you the cerebrospinal fluid viral load. So again, it's the level of HIV RNA, the number of copies in a milliliter of fluid. And what you can see here is that even as early as about 5 days after infection, we've been able to detect HIV in the central nervous system using this method. So what does this really tell us? Okay, the virus is entering the nervous system. Does that actually incite any kind of problem? And what we've seen, we've used multiple measures. This is a measure called CSF neopterin. Neopterin is a soluble biomarker of macrophage activation, and looked at the level of the CSF neopterin. And again, these individuals were recruited very early after HIV transmission. So the x-axis here is the number of days after they acquired HIV. And what you can see is that, and the dotted line is the normal value of neopterin in a healthy patient with no neurologic issues. So what you can see is that most of the individuals have an elevation suggesting myeloid immune activation in the central nervous system very, very early after they acquire HIV. What about this question of when local infection of the CNS cells actually begins? And these are experiments that we've been able to do, again, by comparing the sequences of viruses detected in individuals in early infection between the viruses in the blood and the spinal fluid. And here are a couple of more simple phylogenetic trees that are just showing a single time point in 2 different individuals. These are both individuals where we obtained CSF and blood in the first year of infection and compared the sequences in the spinal fluid and the blood. And in both of these examples, we found compartmentalization, indicating that there seems to be a local cell in the central nervous system producing this virus that looks different than the viruses that are just mixed naturally between the blood and the CSF. In this study, we looked at about 78 paired samples, and we found that in the first year of infection, about 13% of them demonstrated this type of compartmentalization, suggesting that there was a local cell producing the virus. We were able to capitalize on the fact that the natural history study obtained paired samples of plasma and spinal fluid over a number of years in individuals who didn't start treatment. Since they didn't start treatment, they basically had ongoing viral replication, and you can see the levels of the virus in the blood over the first couple of years of infection, and then in the same individuals, the level of the virus in the spinal fluid, indicating that the spinal fluid virus wasn't just high in the beginning, but actually continued to be present over time in all of the individuals that we assessed. This also allowed us to then compare over time the sequences between the blood and the spinal fluid. And we found, again, evidence of local infection. So in this case, we took samples from the CSF in blue and compared their viral sequences to samples of the plasma in red, and showed that, in fact, starting as early as 165 days after the first exposure to HIV, this individual had unique viruses that were found only in the nervous system, and that these seem to locally evolve over time over the first 2 years of infection. So this really, we felt, sort of showed that the virus is able to establish a local infection and locally evolve in the early stages of infection. So I think really the important question clinically for patients living with HIV now is, what happens when you take someone like this and put them on treatment? And so the rest of the talk is talking about pathogenesis and persistence during treatment. And this is an imperfect thing because, of course, not everyone gets on treatment and not everybody gets on treatment and consistently stays on treatment. But the standard of care now is to treat everyone with HIV as soon as we find out they have infection. But what we don't know is whether or not when we're treating people with HIV, there's persistence of some of the pathogenic mechanisms, particularly in certain compartments over time. And it's been known for a long time that certain compartments, and particularly the central nervous system, may have some barriers to complete treatment with antiretroviral therapy. One is that there is reduced penetrance of some antiretrovirals into the nervous system. There may also be different immune responses in the different tissue compartments of the body that maybe don't as effectively address remaining activated cells or infected cells. And the other thing is that there may be different target cells, of course, in different compartments, and that some of our medications may not be as effective in certain cell types. And I think that when we think about ongoing pathogenesis in the CNS, there's sort of three key things. One is whether or not there's just persistent and smoldering inflammation, whether the changes that have been incited in the pre-treatment phase are somehow propagated and don't completely resolve with therapy. Another question is whether or not there's actually ongoing viral replication in the central nervous system that is somehow not necessarily being controlled by antiretrovirals. And finally, are there infected cells that may be latently infected? The DNA has actually integrated into the host cell, and that is not being eliminated by antiretroviral therapy, which doesn't address integrated viral DNA. And those are some of the potential contributors. How many more minutes?
3.
Okay, all right. Well, I'm going to, I think I'm going to just go through this quickly. So one thing is there's a lot of evidence for ongoing CNS inflammation. This is PET imaging evidence, CSF evidence, and some autopsy evidence, suggesting that the microglial activation does not resolve with treatment. There's also evidence that there can be viral replication during suppressive therapy. This is a clinical example of someone who had a suppressed viral load over many years, and then came in and we found that he still had detectable virus in his spinal fluid. This patient actually had resistance genotyping of his virus that suggested that he actually had localized resistance in the central nervous system that was not present in the blood. And he had resolution of signs and symptoms with treatment. So you can have ongoing viral replication, and some very, very elegant work by Sarah Joseph and Ron Stronstrom has suggested that this escape virus that's persisting despite treatment may be coming from macrophages. There's also suggestion that there's ongoing detection of HIV DNA, and this is a study where we looked at the cell-associated component of the spinal fluid and found cell-associated RNA and cell-associated DNA in samples of people on long-term suppressive therapy. And this actually associated with cognitive performance. People with detectable DNA had worse performance. This is a snapshot of some single cell work that we've done in cerebrospinal fluid, where we've identified unique cell populations in the CSF, shown in blue, compared to the blood. And in fact, we identified a cell type that looked like it was a unique cell type that hadn't been previously recognized, that has many features of a microglial cell type. And further studies have done, being again, this is with collaborators and Scorch, have identified CSF HIV transcripts in people on suppressive therapy, in this case, primarily in the central memory T cell compartment. So one final way that we can look at the brain is to look at, as I mentioned, PET imaging. And this is some early work that we've done in a preliminary nature, identifying that there's actually reduced synaptic density in the brain. So I mentioned neuronal cell loss, but there seems to be reduced synaptic density as well in the brains of people on well-suppressive therapy. And this associates with cognitive impairment. This is interesting because synaptic density is potentially reversible. So I wanted to end by just showing a snapshot of the preliminary data that we have developed in our Scorch consortium. So this is from the Y-Scorch, which is focused on HIV as well as opioid use. And we've generated data from control and HIV brains. The HIV brains are from the NNTC, and identified actually variety of differences between the cell types in the insular cortex, prefrontal cortex, and the ventral striatum, associated, interestingly, primarily with some of the immune cell subtypes. And so that's something that we're now actively investigating. To try to summarize the pathogenesis of HIV in a complete, very, very broad way, I think that there's a lot of evidence that there's early injury in the CNS, even without the development of dementia. Much of this can be improved or I think ameliorated by starting antiretroviral therapy. But there are lots of these different features that are co-infections, comorbidities, as well as substance use, which may also be contributing to brain changes that can be cognitively affecting patients. And one of the things that I think we're so interested in looking at in Scorch is how this affects the brain outcomes. This is a sort of nice summary journal issue of AIDS a few years ago that focused all on the central nervous system. We have nice articles by a lot of our collaborators who have expertise in this area. And I'll just say that I think some of the key unanswered questions relate to which cell types and CNS tissues harbor persistent HIV. We haven't looked at the meninges, we haven't looked at the choroid plexus, and obviously there's emerging data that potentially T cells are also detected in the CNS and in people with HIV in the brain. And this may be another site of persistence. And I think that the question that's really key in Scorch is how comorbidities and substance use contribute to this HIV pathogenesis and persistence. So I think the Scorch mandate is to try to tie some of the things that we've learned from studies in humans, where we can't look directly at brain tissue, with what we can learn from looking at autopsy brain tissue. And I'm sorry to race through that, but I do appreciate your attention. And I want to just acknowledge all the study participants, the donors, of course, as Howard mentioned, who have been so generous to give their brains to, for us to be able to do this research, to all of my collaborators, and to our sources of funding. Thanks so much.
Yep, thanks.
Thanks a lot, Serena. We have time for one question. Is there any study on CSF antibody responses, including those that are HIV specific, as well as glycan-specific autoantibodies?
Well, that's wonderful. So there's a beautiful paper from Donica Buzda and colleagues that I'll find the reference and put it in the chat, or Donna can do that, that is recently looking at CSF antibody responses and actually specifically looking at the glycans. The work that I'm most familiar with that I've been involved with was actually looking at CSF HIV-specific antibodies in the blood and the CSF, and finding that in people with chronic infection who are on therapy, there's widespread CSF antibody detection. But in people who start treatment early, not even as early as our Thai colleagues, but if you start treatment early and within the first year or so of infection, there's actually a much, much lower detection of CSF antibodies, despite detection of antibodies in the blood. So what that means isn't exactly clear, but it may suggest that there's maybe less antigen present in the central nervous system in people who start treatment early.